Accuracy. Review procedure used to establish the accuracy of test result and introduce some statistical concepts that describe how close a measurement is to the true value. Distinguish between precision and accuracy. Describe how calibrator value are assigned. Identify the rule of proficiency testing, external quality assurance, and quality control testing program ensuring the accuracy of test results. Laboratory tests must meet precision and accuracy standard. Accuracy closeness to a true value depend on a valid calibration process. Calibrator value assignment is linked to certified reference material, recognized reference method, or continuous process that provide traceability. Laboratory use quality control and proficiency testing to monitor the precision and accuracy of test methods. Precision and accuracy. If the true value for analyte is represented by the bull eye on a target, then accuracy is equivalent for hitting the bull eye every time a measurement is made. Inaccuracy is opposite and means missing the target or bias. Accuracy of analytic methods can be described using the concept of precision and bias. Precision is the reproducibility of a measurement. Imprecision is the opposite, meaning increased variability in the measurement. Bias is the deviation from the true value, also known as target value. And this is the relationship between precision and accuracy. Here, imprecise and inaccurate, both imprecise and inaccurate. Here precise but biased, inaccurate. Here accurate and precise. If a sample is divided into several aliquots, laboratory for splitting a sample into a smaller portion and each aliquot is tested for the amount of analyte in it. The result should ideally be the same for all aliquots. This is rarely the case because of the inherent variability of any test method. Closer the value or each other, the more precise and reproducible the methods. A panel presenting the precision is values that are widely scattered in panel B, in panel A, and the panel B and the panel C. In panel A represent imprecision with values that are widely scattered. In panel B and C, the methods are equally precise, reproducible, but the results in panel B are far from the true value, biased, while the results in panel C are close to the true value, accurate and precise, reproducible. Precision is typically a function of the analytic method that is being used. Precision reflects the innate reproducibility of signal generated by the test solution and the stability of the analyzer used to measure that signal. Standard deviation is a measure of precision and reflects the spread of value from the mean average value. Coefficient of variation is a standard deviation expressed as a percentage of the mean. Precision is described quantitatively by the standard deviation. Standard deviation is calculated using the mean value average of all test value and the deviation of each measurement from mean. The standard deviation reflects the spread of value and is often represented by the sample STD or sigma. A test with a mean of 10, the standard deviation of 1 is more precise than a test with a mean of 10 and standard deviation of 4. The mean and standard deviation can be expressed as 10 plus minus 1, where 10 is mean and 1 is the standard deviation. Precision can also be expressed as percentage of mean using the following formula: 100 multiplied by standard deviation divided by mean. The example, the standard deviation is 10% of the mean when precision is expressed as percentage of the mean. The value is termed coefficient of variation. here frequency number of times each value was obtained for the analyst, and here the measured value. The green line shows greater precision than the value. The blue line mean 10, standard of deviation 4, and coefficient of variation 40%. In green line, the mean 10, standard of deviation 1, coefficient of variation 10%. Bias is typically a function of the calibrator process. Calibration is the step that links the magnitude of an optical, electrochemical, any analytical signal to a specific amount of analysis. The accuracy of calibration process is dependent on the value that are assigned to the calibrator. Bias is usually described as a percentage reflecting the difference between the measured value and the true value. For example, if the target value is 50 and the measured value is 45, the bias is 5% out assigned value to calibrator. Assign value to calibrator because getting the right answer for the true value is dependent on the correct interpretation of the analytical signal. The validity of the calibration curve is a critical component of the test accuracy. Assignment of values to calibrator relies on a process that links the value to some agreed upon standard material or reference method. This is termed traceability. Two typical approach to calibrator preparation: use of the primary reference material and method, or use of consensus method and material. Use of primary reference material and methods. If an analyte can be isolated in pure, well-defined form, the use of primary material and methods is usually chosen as the basis for calibration. Assignment of value to calibrator begin with identification of the substance that will serve as gold standard for any analyte. For a simple substance such as calcium, a particular form is chosen to be the primary reference material, perhaps calcium carbonate, calcium phosphate. Then the actual amount of the calcium chosen material is determined. This determination is performed using a primary or definitive reference method such as atomic absorption in case of calcium. The preparation of primary reference material and the assignment of reference value to them are specialized activity. Reference material are typically prepared by government agencies or professional organizations such as National Institute of Standards and Technology, NIST, American National Standard Institute, ANSI, World Health Organization, WHO, Clinical and Laboratory Standard Institute, International Federation of Clinical Chemistry, Institute for Reference Material and Methods, National Institute for Biological Standards, the Joint Committee for Traceability in Laboratory Medicine. Material reference methods and laboratories that maintain reference method database may be accessed in this website. Primary reference material are too costly and often not suitable to be used as calibration in clinical laboratory. They may be insoluble in biological matrices, body fluid sample, or they may be in chemical forms that differ from those present in the biological sample and therefore unable to be detectable by the method used in clinical laboratory. Secondary reference material or materials that are more suitable for analysis by typical clinical laboratory methods are used instead. Their values are assigned by comparison with the primary reference material using an analytic method that is robust enough to measure and compare the analytes in both the primary and secondary material. The primary reference material serve as the calibrator. The primary reference material serve as the calibrator to assign a value to the secondary material. Secondary reference material are prepared in matrix solutions that resemble the actual patient specimen, serum, plasma, urine. Those materials are commutable that is, provide an analytical response similar to that of the actual patient specimen. Commutability can be confirmed by testing reference material and fresh patient specimen together using 2 or more routine field methods. If the reference material is commutable, the result from the field method should recover the target value assigned by the reference method, and the analytical response should be consistent with that of the fresh patient specimen. Calibrators for clinical lab tests are often prepared in solutions that resemble a patient sample, blood serum. The value of an analyte in the calibrator solution is established by the comparison to the secondary reference material. This comparison and the assignment of the calibrator value is done by the manufacturer of the reagent and test equipment. The primary reference gold standard material serves as the calibrator to assign a value to the secondary reference material, which in turn is used to assign calibrator value for use within the laboratory. This unbroken chain of connections is termed traceability. And example, each calibrator value is traceable to some identified standard material. Illustrate the traceability chain of the cholesterol as standardized by the cholesterol reference method laboratory network, an international network of laboratories that provide certification to manufacturers of equipment and reagent for measurement of the cholesterol. And here the cholesterol reference methods, definitive methods, isotope, dilution, mass spectrometry, primary reference material, and the secondary methods, secondary reference materials, select measurement procedures, calibrator, use of constant value. For many analytes, the preparation of pure reference material that can serve as a primary standard is impossible. Analytes such as protein often have many different forms that may be present in different amount in different patients. Thus, it's difficult to identify one form of the protein as an ideal reference material. Other analytes, like bilirubin, are inherently unstable and break down when exposed to light, air, or when separated from other stabilizing molecules in solutions. Enzymes often lose their enzymatic activity if isolated in pure form. For these types of analytes, no suitable primary reference material can be prepared. Instead, values for these analytes are traceable to a constant value based on approach that have been established by agreement among laboratory professionals. Hemoglobin, HbA1c, the most important test for long-term diabetic control, is an example of the test that is standardized by constant process. When hemoglobin is exposed to glucose, it can undergo a modification in which glucose molecule chemically attached to the protein. The product is called glycohemoglobin or glycated hemoglobin. Glycated hemoglobin, since this attachment can occur at any of several different sites on the hemoglobin molecule, the result is heterogeneous mixture of unmodified hemoglobin and various glycohemoglobin molecules. Values assigned to the test for HbA1c are based on the methods that was used in large clinical trial called diabetes control and complication trial. That trial identified target value for HbA1c to achieve optimum control of diabetes. Since clinical interpretation of the test result is based on the outcome from that trial, the clinical utility of the patient test result is tied to how well it matches the result from the method used in the trial. The trial identified target values of HbA1 to achieve optimum control of diabetes. Since clinical interpretation of the test result is based on the outcome from the trial, the clinical utility of the patient test result is tied to how well it matches the result from the method used in that trial. That method, a high-performance liquid chromatography method using Biomex resin, has been adopted as a consensus method by the National Glycohemoglobin Standardization Program. Illustrate how results from the method used by clinical laboratory are traceable to the value obtained by the consensus methods from NGSB laboratory. The consensus method is used to calibrate secondary methods in specified certified laboratories. Manufacturers as well as clinical laboratories can compare the result from their methods with the result from a secondary lab to confirm accuracy. Manufacturer use this result to assign appropriate value to calibrate, so patient sample will give results comparable to the consensus method. All comparisons are carried out on blood sample collected from diabetic and non-diabetic donors. National Glycohemoglobin Central Primary Reference Laboratory Reference Material Secondary Reference Laboratories Fresh blood samples and user test methods. Verify accuracy by split sample testing with secondary reference methods. An alternative reference measurement system for standardization of HbA1c has been adopted by International Federation of Clinical Chemistry. In this approach, two purified forms of hemoglobin are isolated and used for calibration. One is unmodified and carries no glucose molecule. The other carries a single glucose molecule attached to the amino acid valine at the amino end of one of the beta chain of hemoglobin. A series of standard solution is prepared by mixing different proportion of these two forms of hemoglobin. The results are expressed as ratio of the modified form to the unmodified form. In addition, the IFCC has developed two equivalent reference methods: liquid chromatography, capillary electrophoresis, and liquid chromatography mass spectrometry. Results from the IFCC NGSE methods can be interconverted using a validated master equation. The NGSE and the IFCC work with each other to ensure continuous standardization of HbA1c. IFCC work with each other to ensure continuous standardization of HbA1c method. Interconversion of the result between percentage of HbA1c and millimole per mole HbA1c and clinical acceptable performance of commercial test methods.